Hello everyone, thank you for joining me today for this talk entitled An Innovative Product That Goes Beyond the Concepts of Pre, Pro and Postbiotics. As you may know, I am Marco Ruggiero, I hold an MD and PhD, and this product I talk about is called OneVar, the one and only panbiotic. And the talk and the product are made exclusively for Danabiologics and Mr. Choi-Yuk. Before we start, please allow me to introduce myself. I obtained my MD in 1980 in Florence, Italy. I have a PhD in molecular biology and a specialization in diagnostic radiology. As a young postdoc, I worked at Burroughs Wellcome in 1984 in North Carolina, USA, and later as visiting scientist at the National Cancer Institute of the NIH in Bethesda, Maryland, since 1987. Later, I was appointed professor of molecular biology at the University of Firenze, or Florence if you prefer, in Italy, in 1992, where I worked until 2014. I am the founder of Silver Spring, a Swiss company specialized in research and development of innovative approaches and products, including OneVar, the product I'm talking about today. Over the course of my scientific career, I published more than 240 scientific articles, co-authored, among others, by scientists such as Eduardo Lapetina, Stu Aronson, Jackie Pierce, Peter Duesberg, Henry Bauer, the late Dr. Jeff Bradstreet, Dietrich Klinghardt. One of my articles was sponsored by Nobel laureate Sir John Vane. My current research interests are in immunotherapy, oncology, and neurosciences. Also, before we start, please remember that no information in this talk is presented as medical advice. Standard of care for every disease must be followed, as well as rules and regulations established by health authorities. I wish to disclose my conflict of interest that is quite evident here, because I have designed a number of protocols and approaches to different diseases, and I developed the product, OneVar, and the approaches mentioned in this talk. I wish to thank Mr. Choi-Yuk of Danabiologics for his priceless support. This talk is for him, this product is for him and his company. And also I wish to thank all those who will watch this video for the attention that they will want to give to this presentation. Let's start with some definitions. Prebiotics, what are they? They are nutrients that qualitatively change the composition of the gut microbiota by providing food, if we may call so, that promotes good bacteria growth. Probiotics, these are the microbes, the microorganisms themselves, like lactobacilli, bifidobacteria, and so on. Postbiotics, these are the metabolites of probiotics, or, if you wish, the components that result from the probiotic activity, like fermentation. As probiotics consume prebiotic fibers, the result of that fermentation or consumption is what is known as postbiotics. In short, prebiotics are the food, probiotics are the microorganisms themselves, and postbiotics are the results of probiotics consuming that food. OneVar is something different, because it combines all these properties, and therefore it can be defined as the one and only panbiotic. This is a new term for a new product, a term that indicates a product that encompasses all. Pan is a prefix from the Greek pan that means all, or of everything, or involving all members of a group. A new name for a new product. OneVar, the one and only panbiotic. So, what is OneVar? What makes OneVar? Essentially, there are 5 components, because OneVar is an original entire ecosystem with self-assembling supramolecular structures. The components are zinc, an essential mineral, a proprietary culture of prokaryotes and eukaryotes with their theater of activity, a long-chain sulfated polysaccharide, LCSP, of vegetal origin, endowed with GC-MAF-like activity. More on GC-MAF later. And then there are two botanical extracts. The first one is a botanical extract with known anti-inflammatory, immune-modulating, anti-cancer, antioxidant properties. Then there is a second botanical extract that contains natural free phenolic compounds, fibers, macro and micronutrients with known anti-inflammatory, immune-modulating, anti-cancer properties and known to rebalance sugar and lipid metabolism. As I said, more on GC-MAF in the next slides. If you do not know what GC-MAF is, I recommend that you take a look at this article that was published as a special report in a famous British magazine called What Doctors Don't Tell You. And actually, this is the cover of the magazine. So it had the cover of the magazine, and it is called GC-MAF, the superhero. Well, if you want to go to PubMed, which is the official website of the US government, the National Library of Medicine, the National Center for Biotechnology Information, and you just look for GC-MAF, you will find, among other things, that is proposing cancer immunotherapy in viral conditions from COVID-19 to HIV, and also in neurodegenerative diseases. For example, this article deals with multiple sclerosis. And I have published a number of scientific articles on GC-MAF that, again, you can retrieve from PubMed. Just look for Ruggiero GC-MAF, and you find a number of articles where I describe a number of effects of this molecule, as well as I explain its mechanism of action. Another important point is that 1VAR is designed according to a proprietary, highly innovative procedure that optimizes molecular interactions between its 4 or 5 components, so to increase their respective activities. In other words, 1VAR is not the result of the addition of the activities of its 5 components, but the result of the multiplication of the activities of its 5 components. In other words, if the biological activity of each individual component of 1VAR were 10, the overall activity of the self-assembled components would be 10 by 10 by 10 by 10, 100,000, rather than 10 plus 10 plus 10 plus 10 plus 10 that makes 50. In other words, its activity is 100,000 versus 50. This synergism is obtained at the molecular level through carefully calibrated interactions associated with the distribution of charges on the surface of molecules, leading to self-assembly of entirely original supra- and super-supramolecular structures. So let's talk about these 5 components. And let's start with zinc, an essential mineral. Zinc is a trace mineral that is essential for many of the body's normal functions. It is involved in a wide range of processes, including immunity. It is important for the development and function of the immune system. It helps to protect the body against infection. It is essential for wound healing and also for repair in general. Helps to promote the growth of new tissue and the formation of scar tissue. For the sense of taste and smell, it helps to produce the enzymes that are needed to detect flavors and smells. It is involved in cell division. It helps to repair damaged cells and create new cells. Protein synthesis. It is essential for protein synthesis. It helps to build and repair proteins in the body. It's involved in metabolism. It helps to break down food and release energy. Eye health helps to protect the eyes from damage and improve vision. Male fertility helps to produce a sperm and improve sperm quality. Pregnancy helps to support the growth and development of the fetus. And of course, if you have deficiency of zinc, you have deficiency of immunity, and the immune system is weak. and this makes people more susceptible to infections. And if you have deficiency of zinc, of course, wound healing doesn't work as it is supposed to do. Taste and smell are impaired. Cell division slows down. Any repair at level of cells is slowed down or it is deficient. Protein synthesis is impaired and muscle wasting may occur. Zinc deficiency can slow down metabolism and lead to weight loss. Zinc deficiency can damage the eyes and lead to vision problems, can reduce sperm count and motility, and decrease male fertility. And in pregnancies, zinc deficiency can increase the risk of complications during pregnancy, such as premature birth and low birth weight. You may find many supplements containing zinc. So 1-var is not the only supplement containing zinc or functional food containing zinc, but in 1-var, zinc is particular because it is arranged in a supramolecular complex with other components, so to achieve optimal absorption, pharmacodynamics, and intracellular efficacy. So you do not find this in any other supplement containing zinc. This happens because zinc is a metal and it has a valence of +2, so this means the zinc ions are positively charged. And zinc ions are important for many biological processes, as we have seen before. They are involved in the formation of proteins, DNA and RNA. They play a role in wound healing, immune function, and taste. We have already seen these, and these are the ions that are responsible for these functions. Thanks to its positive charge, zinc in 1-var is bound to the long-chain sulfated polysaccharide that is a highly negatively charged molecule, as it will be described in the following slides. This binding affects the structure and function of the LCSP. For example, zinc binding increases the viscosity of the LCSP, and this makes the LCSP more effective. So there is a double synergism. The binding favors zinc, and zinc also favors the efficacy of the LCSP. Also, it is very important to notice that zinc affects the degradation of the LCSP because it inhibits the enzymes that break down the LCSP. And this can help to protect the LCSP from damage and improves its GC-MAF-like activity. So this is just a very simple representation of how zinc, of course this cartoon is not in scale, zinc is not this big in comparison to the LCSP. So, but this is just to give you an idea of the molecular interaction between the zinc ion and the negatively charged LCSP that in turn is bound to other components of 1-var. In this case, for example, the polyamines that are produced by the Saccharomyces boulardii. So this cartoon depicts the interactions and the self-assembly of zinc with the LCSP and the polyamines produced by the Saccharomyces boulardii. And this leads to the formation of a multimolecular complex in the aqueous ionic environment of the intestine. Let's move to the second component of 1-var. A proprietary culture of prokaryotes and eukaryotes with their theater of activity. Of course, the precise microbial composition of the proprietary culture of prokaryotes and eukaryotes is protected by intellectual property, so I cannot give you all the details, but let me just bring an example, the Saccharomyces boulardii. That is one example of the eukaryotes that compose the proprietary culture, and let me give an example of their health supporting properties. The S. boulardii shows many activities. Let me just list a few of them: antimicrobial, antitoxin, anti-inflammatory. But most important in the context of 1-var is the regulation of immune responses, among which it triggers the activation of complement and migration of monocytes and granulocytes, thus synergizing with the natural GC-MAF of 1-var, that is the LCSP. Enhances the mucosal immune response, enhances the systemic immune response, contributes to earlier production of interferon gamma and interleukin-12, stimulates regulatory T cells, modifies migration of lymphocytes in a chronic inflammatory bowel disease model, modifies, that is, improves lymphocyte adherence to endothelial cells, improves cell rolling and adhesion. In other words, it has a number of functions that support the immune system. What is the theater of activity that I have mentioned more than once? Well, this is a novel concept because what is important is not only the probiotics, it is the microbes, bacteria, or eukaryotes like the S. boulardis and eukaryote, but it is important what they make, what they have together with them, that are the microbial structure elements like protein and peptides, lipids, polysaccharides, and their nucleic acids like DNA, RNA, and also the mobile genetic elements like plasmids or the phages that they carry with them, also called bacteriophages, that compose the human virome that is has been recently described, and it is essential for our health. And also there are relic DNA. And then there are the environmental conditions that contribute to these complex, well-defined habitat that has distinct biophysiochemical properties. And then there are the microbial metabolites and the postbiotics that are those molecules that are produced by the microbes, signaling molecules, anorganic molecules, GCMAF produced by the microbes. So in one VAR, we have all of these. We don't have only the probiotics, we have the prebiotics, the postbiotics. This is why one VAR is called the one and only panbiotic. Let's move to the third component, a long-chain sulfate polysaccharide, LCSP, of vegetal origin endowed with GCMAF-like activity. This is just one of the many repetitive units of these polysaccharide. I don't want to go into the biochemical details because I know that they could be very boring, but just let me tell you that even if you are not a trained biochemist, if you look at the LCSP and at this other molecule that is chondroitin sulfate, I think that you can recognize that these two molecules are very similar. As a matter of fact, LCSP shows a high density of negative charges that are represented by the sulfate group and also the carboxyl groups that are uniformly distributed on the molecular surface. Now, why do I focus on chondroitin sulfate? What has chondroitin sulfate to do with one VAR? Is there chondroitin sulfate in one VAR? No, the answer is no. There is LCSP. But, as I have shown before, LCSP is very similar structurally to chondroitin sulfate. And this is important because I demonstrated, together with my colleagues, Dr. Stefania Pacini, and a great German doctor, Dr. Heinz Reinwald, in 2016, we demonstrated that chondroitin sulfate is responsible for the biological effects of GCMAF. You can retrieve this article from PubMed, the official website of the United States government, more precisely of the National Library of Medicine of the United States of America. How do we know that the LCSP has GCMAF activity? Not simply because it looks like a chondroitin sulfate. There have been experiments on colon cancer cells, and as we can see, there is a dose-dependent inhibition of the alpha-nagalase from colon cancer cells by this LCSP. Please notice these are micrograms, so very tiny amounts. So it's very effective in its GCMAF-like activity against nagalase. But LCSP does more than simply being or having GCMAF-like activity, because it has a number of other activities that contribute to the health-supporting properties of one VAR with particular reference to immune modulation. It has been demonstrated that it enhances the phagocytic and secretory activity of macrophages, induces the production of nitric oxide, more about this later, and cytokines like TNF-alpha, IL-1, interleukin-1, and interleukin-6. And to this point, it is worth noticing that I demonstrated that nitric oxide is responsible for the cancer-killing effects of GCMAF, as it will be shown in the following slide, where we will see how activated macrophages surround and kill human breast cancer cells thanks to the production of nitric oxide. So let me guide you through these slides. These were taken under a microscope. So these are big human breast cancer cells. These are other big human breast cancer cells. These little dark blue dots are macrophages. So you see how many macrophages they surround the cancer cells. So let's go to a higher magnification. So this is one human breast cancer cells, and these are macrophages all around it and also on top of it. And let's magnify even more. Again, one big dying human breast cancer cell surrounded by these little dark blue dots that are macrophages. And this is probably the most impressive one. This is the highest magnification. Can you see here the macrophages is drilling a hole inside the cytoplasm of the cancer cell. And this is the nucleus of the cancer cell, and the cancer cell is dying of apoptosis. That is, its DNA, it's all fragmented. So you see its DNA has all these little dots. It means it is all cut in pieces. And here you can see other macrophages that are drilling holes in the cytoplasm. This is the big cancer cell. So they're killing the cancer cell. How do they kill the cancer cell? Thanks to the production of nitric oxide. Nitric oxide is like a bullet that the macrophages use to shoot and kill the human breast cancer cells. And this is a common mechanism. I was the first to demonstrate and publish this almost 10 years ago. And now let's move to another component, the first botanical extract that has known anti-inflammatory, immune-modulating, anti-cancer, antioxidant properties. It has been shown to have anti-cancer properties. It can prevent the development of certain types of cancer, such as lung cancer, breast cancer, and colon cancer. It may help with weight loss. It can help boost metabolism and increase fat burning. As far as brain health is concerned, it has been shown to improve brain function. It can improve memory, learning, and cognitive function. It may also improve heart health. It can help reduce the risk of heart disease, stroke, and high blood pressure. And it's also a good source of antioxidants. Antioxidants help protect the body against the damage from free radicals. And also contains a number of hydrophobic compounds, which are compounds that are not soluble in water. These compounds include catechins. These are polyphenols. They have a number of health benefits, including cancer prevention, weight loss, and improved brain function. Flavonoids, other type of polyphenols that have antioxidant protection and improved heart health. Triterpenes or triterpenes that are types of lipids that have anti-inflammatory, anti-cancer properties. Lignans, these are plant compounds, have a number of health benefits, antioxidant protection, improved cholesterol level. Now, the hydrophobic compounds in this first botanical extract that composes 1-var, these are responsible for many of its health benefits because these compounds are able to penetrate cell membranes. So they go inside the cells and protect the cells from damage. And they also have anti-inflammatory and antioxidant properties. And then there is a second botanical extract that contains natural free phenolic compounds, fibers, macro and micronutrients with known anti-inflammatory, immune-modulating, anti-cancer properties and known also to rebalance sugar and lipid metabolism. This second botanical extract exerts multiple functions in the context of 1-var. On one side, it contains a number of bioactive molecules that interact with each other and with the other components of 1-var in a synergistic way, potentiating each other's efficacy. On the other side, it confers prebiotic properties to the formula of 1-var. And finally, it serves the purpose of a backbone. That is a docking structure where the other components of one VAR are assembled in a supramolecular configuration. Because of these features, it is abbreviated in BBSS, Botanical Backbone Supramolecular Structure. Now, again, I don't want to be boring with these molecular structures, but just to give you an idea of how they interact. This is the long-chain sulfated polysaccharide, and these are the electrostatic interactions with the BBSS, the botanical extract. And then we go to much more complex structures that self-assemble in our bodies when we ingest the one VAR. And so this is a very complex triple bilayer with a zinc atom at the center. Of course, again, this is not in scale. It's just to give you an idea. And I don't want to go into many biochemical details because I know that molecular biology can be very boring. I've been teaching molecular biology for some 20 years. I know it can be boring. I don't want to bore you to death, but I just want to tell you that we know perfectly well the molecular interaction between all the components of one VAR, and this is how one VAR has been designed. Now, you may say, oh, well, fine, all these may be interesting or maybe not, but is it working? How can we assess the efficacy of one VAR on the immune system? How can we be certain that it is working in a repeatable, objective way? Well, as I have said before, and as it has been demonstrated since 1997, so many years ago, when macrophages are activated, they release nitric oxide, a molecule that is essential for the killing of cancer cells and cells infected by viruses. As this old article writes, sustained production of nitric oxide endows macrophages with cytostatic or cytotoxic activity against viruses, bacteria, fungi, protozoa, helminths, and tumor cells. So this is a fact. When macrophages are activated, they release nitric oxide. So this is how we can demonstrate that one VAR works, by looking at the nitric oxide that is released by the macrophages. And how can we do this? Well, let's remember nitric oxide does many things. One of these things is to induce vasodilation. So now the blood vessels under the influence of nitric oxide become larger, wider, and there is increased blood flow. As a matter of fact, nitric oxide is used because of its efficacy on vasodilation, and this has been known for centuries, I would say. So now we have to find macrophages in our bodies, and we have to assess whether they release nitric oxide or not. Where do we find macrophages in our bodies? Well, essentially everywhere, because they are circulating. When they circulate, they're called monocytes. When they are resident in tissues, they're called macrophages. So you find macrophages in all tissues, but where are they highly concentrated? In the adenoid, in the tonsils, in the thymus, if we still have thymus, in the bone marrow, in the lymph nodes, but also, and this is important, in the spleen. So in our spleen here, on the left, we have a high concentration of macrophages. I mean, this is a common knowledge. Everybody who has studied anatomy or histology knows that there is plenty of macrophages in the spleen. And we can easily use this, study the spleen using ultrasonography. This is a common ultrasound machine. This is a picture from my archive. So this semi-lunar area here is the human spleen. This is diaphragm, the upper pole of the spleen, the lower pole of the spleen. So this thing here looks like a half moon, or like a sickle if you prefer. This is the spleen. And we can study the spleen with an application of the ultrasound machine that is called echo-color Doppler, that studies the blood flow. So let me show you the next couple of slides. They show you how release of nitric oxide from activated macrophages can be studied in the spleen. So this is before assumption And you see only a few blood vessels, not much blood circulation in a normal spleen. But after the assumption, look at this, look at all these colors. Here, both the cortical vessels and the hilar vessels of the spleen, they are vasodilated, a lot of blood flow. So this indicates that there has been a huge release of nitric oxide by the activated macrophages. This is another slide, enlargement. So again, now you can recognize that this thing here is the spleen, and these patches of color indicate the flow of blood. Red is when it moves in one direction, blue is when it moves in another direction. We use the acronym BART, blue away, red toward. So it's not venous and arterial. It depends on the direction of where the blood is going, looking at the probe. But don't be bored by these technical details for radiologists. Also, we can assess the efficacy of 1-VAR using the immune status questionnaire. This is a questionnaire that was developed about 4 years ago in 2019 by these researchers. You can go and take a look at the original article. And this questionnaire can be given to people who are taking 1-VAR, and you can see numerically the improvement of their immune status by looking at this. And we are collecting data in this regard. Now, here we talk about Basel, Switzerland. And this leads me to another point of my talk, because 1-VAR is made in Switzerland. So it's a product of Switzerland, and I think that you all recognize that Switzerland is known all over the world for the highest quality of its products. This is another map of Switzerland. Our company is located in this canton of Switzerland, and our manufacturing plant is located up here. But you know, it's a small country. It only takes about 3 hours to go from the seat of our company to our manufacturing facility. So we go there quite often, and I'm always there supervising the production of the most important products of ours, including, of course, 1-VAR. This leads me to something that I don't know how familiar you are with, the Demeter certification, because our manufacturing plant is certified by Demeter. Who is Demeter? What is Demeter? Demeter certification is a guarantee for consumers that the products come from biodynamic agriculture. So 1-VAR is manufactured in a facility that is certified by Demeter. And I tell you, very, very few supplements in the world can make this claim, because it's very difficult to have certification by Demeter. Because biodynamic agriculture is a holistic, ecological, and ethical approach to farming, was developed by Austrian philosopher Rudolf Steiner in the 20th century. Demeter certification is the strictest organic certification standard in the world. It goes well beyond the requirement of organic certification by requiring farmers to use biodynamics preparations, which are made from plant, animal, and mineral materials. Biodynamic preparations are used to improve soil fertility, plant health, and pest control. So we are talking about the strictest organic certification standard in the world. And as I say, not many, I don't know any, supplements can make this claim. Why? Because, you know, it's not only important that our health is good, but also the health of the planet, the health of the environment is good. Otherwise, what's the point of having good health if we live in a polluted environment? And Demeter is exactly what it's doing, because the Demeter certification requires farmers to manage their farms in a way that is beneficial to the environment and to the wider community. The farm must be managed as a holistic unit, with the soil, plants, animals, and people all working together in harmony. So in 1-VAR, we have the molecules working together in harmony, synergizing with each other, producing this multiplication effect, 100,000 more efficient than 50, just like the people who make one VAR, work together in harmony with the environment, with the soil, with the plants, with the animals. And for the consumer, the meta certification is a way to ensure that what they are buying is produced in a sustainable and ethical way. And finally, why one VAR has this name? This is not a commercial name. This is a code name. The commercial name designation is different in each different country. My good friend, Mr. Choi Yuk and Danabiologics will decide how to baptize one VAR in their country, in the Republic of Korea or in China or in Southeast Asia or wherever they want. This product is made exclusively for them. But the code name, one VAR, comes from a French region or department of Var. It's a wonderful place in southern France where I found inspiration for such a unique and innovative product. With this beautiful picture, I wish to thank you so much for your time and attention. And of course, if you have any question, feel free to shoot me an email and I shall be very happy to answer any question of yours. Thank you so much.